Thank you very much. We call our speakers back to the panel table, and we would like to welcome two new panelists, Dr. Hiren Patel, Senior Staff Fellow from the Office of Bioequivalence at OGD, and also Dr. Pahala Simamora, Division Director of the Office of Product Quality Assessment in the Office of Pharmaceutical Quality. So we'll do the same thing like we did last time. Any in-house questions, please come up to the microphones. We have such a lovely panel of experts here. They're just waiting to answer your questions. Brave, sir. Yes. I hope you're going to the microphone. Okay, yeah.
Yeah.
All right, question at mic 1, please.
Marie Ducharme, Learning Confirm in University of Montreal. I have a question for Dr. Tsakalozou regarding, first of all, it's a comment, and then the other one is a question. The comment is, you said that the model established a link between systemic and topical concentration. Isn't that an assumption of the model?
It characterized the relationship, right?
Well, it's an assumption of the model. It's built in.
Yeah, it's part of the structure of the model, yes.
Yeah, okay. So I think the sentence is a little bit misleading. I mean, just a comment.
Sure, thank you.
The question is, what is your validation criteria? You know, you showed, you know, validation with data and all of that. What is it?
Yeah, that's an excellent question. So you're asking this question because the performance of the model was not always ideal, right? So the challenge that we typically see in PBPK modeling, and it was very, very obvious with this type of work that I presented here, was that there was a lot of variability in the in vivo PK data. The conditions, the study conditions varied between the ANDAs that were utilized for model validation in terms of the population, the application sites, the doses that were applied, and the conditions. So rubbing, for instance, is a big consideration that unfortunately currently we don't take into consideration into the model. And this is a condition that may impact skin permeation to a great extent. The other challenge specifically for this work is that the application site was not the same across all of these studies. So that introduced variability in our model predictions. The criteria that we used in terms of the internal validation is a 20% difference between the predicted and the observed. We want to be as close as possible when it comes to the internal validation, but for data sets that are used for external validation, there are parameters that come into play in terms of the population variability that we cannot always account into our model, and that leads us to the model performance that you saw here.
Yeah, but what was the criteria? So it was like minus to plus threefold, or what was it?
It was twofold.
Twofold? For the mean?
Yes.
The mean being twofold? Okay.
Yes.
Thank you.
Thank you, Dr. I'm going to get this. Dr. Tsakalozou. I'm just saying thanks. Okay. Any in-house questions? Any? Okay, let's go online, please.
Okay. We have actually another question for Dr. Eleftheria Tsakalozou. How are in silico models being validated for a given drug product and indication? If the same model is being used for another product, what variation will have to be considered?
Thank you. That's another good question. So the models that such as mechanistic models, such as the one that I discussed today, are drug product specific. So they're built utilizing data that are specific to the product of interest, and then they are validated either with IVPT, microdialysis, open flow microperfusion, or in vivo PK data that are also collected after following application of that specific product. So in other words, in the examples that I described here today, for every ANDA, two models were developed, one for the reference standard and one for the test product. The validation is performed across all the available data sets. Obviously, we're looking for data sets that are good quality, that are collected under controlled studies. And against the previous question, obviously there is a variability around those data sets. I hope that I've answered this question. Hopefully I didn't miss anything. Thank you.
Thank you. Next online question, please. We'll just continue with the online unless somebody comes up to the mic.
Next question is directed to Dr. Pahala Simamora. Could you please elaborate on the usefulness of in vitro characterization and in silico approaches to support SUPAC changes?
Thank you. That's such a good question. As we know, SUPAC guidance has been in place since in the 90s, so it's been a while. It's been evolving so far, as Dr. Megan Kelchen presented, all the involvement to date. A lot more we learn about drug product, whether it's simple or complex, and so on and so forth, right? So, and also, as the previous section mentioned about looking at the totality of the data as well. The point is, I think with a strong product development, strong product understanding and process understanding, I think it would make it easy when post-approval we want to make changing to certain thing. Is there like a specific things to do? Maybe not. Maybe it's in a combination of things to look at the totality of the information. I don't know if I hope I answered the question. I know it's quite generic, but compared to the what we have which is currently is the standard for post-approval change. But I think now we have identified a lot more parameters that are more meaningful for to identify to mitigate the risk of changing certain things post-approval. Thank you.
Thank you. Next question, please.
Next question is for Dr. Megan Kelchen. Do I need to get feedback on a proposed concentration in my test product if the level is below what is listed in the inactive ingredient database, or IID?
Thank you for the question, and thanks for the opportunity to clarify this. So the external IID is a great starting point when developing or trying to figure out which concentrations you want to use in your proposed test formulation. Because as I mentioned in my presentation, the IID includes the route of administration for the drug product. So the idea being that your proposed concentration should be below the level that's included in the inactive ingredient database for that route of administration. But as I mentioned, the context of use, so for example, the duration of use or the patient population that product is used on or used in is not listed in the IID. And so what we don't want to do is use a product that is for an acute indication to justify the levels of a product or a concentration that's to be used in a product for a chronic indication. And so while it's not necessarily required, we strongly encourage you to come in with your proposed concentrations and get feedback from the agency. And again, this can be helpful even if you want to do an in vivo study, because we don't want you to do a comparative clinical endpoint study just to find out your proposed concentration is too high compared to what we have in-house. Thank you.
Thank you, Dr. Kelchen. Next question, please.
Next question is for Dr. Hiren Patel. Could you please highlight the criteria for recommending IVPT in the in vitro characterization BE approaches? Also, how can an applicant interact with the agency for feedback and recommendations?
Sure. Thank you for the question. It's an excellent question. As we heard, Dr. Kelchen talked about how the characterization-based BE approach laid out. It based on the complexity of the drug product. For example, and it is, as we heard in the earlier session too, that it's a weight of evidence-based approach. So it is how we systematically mitigate the risk of failure mode. For example, if the drug product is a single-phase formulation, then if the formulation is meet the no significant difference criteria, which is based on the Q1, Q2, have similar Q3, and if you conduct IVRT study, we believe that it systematically mitigate the risk of failure mode. There are certain exemptions. For example, if it has a complex inactive ingredient like white petrolatum, then we may recommend IVPT study, even though it's a single-phase formulation. However, if the formulation is a multiphasic, we typically recommend IVPT study in addition to the Q1, Q2, Q3. However, there may be some exception on multiphasic formulation. For example, if there is no clinical relevance, we may waive the IVPT study. So I hope that answered the question about in which instances we recommend IVPT study. The second part of the question is if you seek agency's feedback either on the utility of the characterization-based approach, I would encourage you to submit product development or control correspondence. Thank you.
Thank you, Dr. Patel. We do have an in-house question, please. Go ahead.
Yeah, I have a practical question. Let's say if you use topical drug applied to skin and for a certain time, and you continue measure plasma concentration on those things, so there's a timing difference beyond how when you stop use drug. When you do modeling to the validation, and how long time you need to take after the drug finish.
That's a question for me.
Yeah.
Okay. Yes. So the model predictions are considered validated for as long as there is observed data to track, to compare those simulations against the observed data. So if you have a PK, an observed PK profile that is 12 hours, but your model is predicting for 48, clearly the part of the model predictions that are not validated against, they're not compared against observed data, are not as credible as the ones that are compared to actual observed data, right? There are, when we are assessing model validation, when we're assessing model performance, we're looking for the overall shape of the curve we're interested in, right? So Cmax, AUC, Tmax, and then additional characteristics that may be relevant to the product. Multiple peaks, maybe.
Can I follow up?
Please.
So in certain cases, some drug, they have a faster metabolism rate. And even when they continue release, you might see drug in skin tissue, but the plasma concentration can go down necessarily to 0. And in that situation, you don't see too much drug concentration in plasma, but you still know drug is releasing and you see in tissue. And a validated modeling is going to be what timing cutoff. And either we should use plasma to do the modeling at all. Just wanted your thought about that. I don't know what's right or wrong, just your thought.
Yeah, the case that you're describing is definitely challenging. It would be very useful in this case to follow a more mechanistic approach, modeling approach, I mean, where there's additional information that goes into the model that can justify, that can support further the model predictions. Then it will also be helpful to show that this same model or similar model or modeling suite is also behaving well with other products. So the uncertainty that is present in your own case is in some way mitigated by accumulating knowledge in other cases. Additional areas like matrices where the model can be validated would also be useful. Additional sources like multiple studies will also be validated, will also be useful. Best practices when we are developing a model, overall best practices present in our guidances should be followed to kind of mitigate, to increase our confidence and mitigate that uncertainty, that risk that we have with the lack of the data. It's a very general question.
Thank you very much for that response. Online, please.
We have an online question for Dr. Tannaz Ramezanli. How does comparing metamorphosis and thermodynamics translate into a patient's or prescriber's concern about similar look and feel of a topically applied dermatologic drug product?
As I shown in the presentation, one of actually our research project focuses on that and to see how the differences in composition and Q3 properties can change the sensorial perception of topical products. So our early results or like preliminary results suggest that we can correlate these sensory attributes with in vitro Q3 tests, and our in vitro Q3 tests are usually more strict to differentiate compared to how the differences will be significantly felt by patients. So eventually, I think if we have differences in the composition of the formulation, and the formulation is not gonna go through the clinical endpoint study where it's tested in patients, there might be alternative ways that we can mitigate this failure modes, and we are working to develop the alternative studies for that.
Thank you. Next question, please.
Next question is for Dr. Tsakalozou. Could you comment on whether the US FDA develops models or methods for assessment of in silico studies, or prefer that the manufacturer do so?
Thank you for the question. It really depends on what is the need that a product presents, an application presents. If an applicant identifies the need for their product to be supported by in silico approach, we welcome this. We will call it alternative B approach, and we will we'll provide the necessary feedback. In cases where we feel that there is a risk with the product, in cases where we want to inform our regulatory policies, such as the guidances, we would conduct internally modeling and simulation. So both.
Thank you. I think we have time for one more online.
This is a general question for the panel, or for anyone on the panel. PSG revisions are a big headache for industry. Therefore, why don't you publish a set of correct studies to begin with, rather than revising them one at a time based on approved ANDAs?
So I'll start and others can chime in. So the recommendations we have for in each PSG is product specific and is recommended based on our evaluation of the data for that product and understanding the complexity of the product. So we definitely cannot and would not recommend a general studies for series of different products, because each guidance or product-specific guidance is specific to that particular drug product.
Thank you. Do you want to squeeze in one more online?
Can I actually add something?
Sure.
I'll note that when we publish PSGs for newly approved drug products, sometimes we want to do some additional research before we publish, say, a characterization-based approach. And so we might publish a comparative clinical endpoint study as part of the PSG to get the guidance out there, to give industry an opportunity to have a PSG available. But there might be some internal research that we're doing, such as like the clascoterone cream that I discussed in my presentation. Thank you.
Okay, I think we'll call it, we're just on 11:45. So thank you to our panel.